Good afternoon, this is Jan Buitelaar. I'm working in Nijmegen at the Radboud University in the Netherlands, and it's my great pleasure to welcome you on this first webinar on the EU AIM study. It's a new initiative, and we try to disseminate new findings from the study, as well as other research on autism. And the special occasion is today, it's the first one, and we have two great speakers. Let's introduce them. It's Will Spooren, connected to Roche pharmaceutical company, and he is the pharmaceutical lead, FBI lead of the whole EU AIMS project. And the other one is Declan Murphy, professor at King's College in London, an expert in neurobiological research, among others in autism, and they together are now still the directors and the coordinators of the whole project. Let me explain shortly the whole procedure. We will have a webinar by Will for about 30 minutes, and then there is opportunity for questions. It's my pleasure, Will, to give the floor to you. Please go ahead.
Thank you very much, Jan. Thank you all who have dialed into this webinar today. The first one, as Jan said, in hopefully a series of webinars that we will have. And today what I will tell you is the high-level overview of the project. So we're going through not so much detail, but so that you can have a sort of a broad view of the whole project, which is quite large actually. And in the next set of webinars, we will go more in detail about our studies and the deliverables and the success that we had. Today's presentation is about developing the discovery framework for autism and spectrum disorders. That is the goal of this project, and you will see how we will try to achieve this. First, about autism. So autism is defined by two core symptoms: social communication deficits and repetitive behavior, and there are a number of comorbidities that can include sleep disorder, anxiety, depression, hyperactivity, attention deficits disorder, a cluster of symptoms that are called irritability, as well as GI and G disorders. So the incidence in autism has increased heavily over the past years, where it was, 10 years ago only a rare disorder. To now it's more frequent than schizophrenia. It's 1 in 68 births. There are very clear gender differences. It's 4 times more frequent in males than it is in females. There's a clear genetic link, and there's basically no treatment for core symptoms, and there are 2 antipsychotics indicated for the treatment of irritability. So one can say there's a high medical need with no therapy available. Now, when Roche committed to this field, now about 10 years ago, it was very clear that there was no framework for developing or drug discovery in autism. So in order to move this field into evidence-based medicines, one needed a concerted effort of key stakeholders to move this field into this evidence-based medicines. In the absence of great progress, a way forward would be a concerted effort, as we know that in Europe, in a private-public partnership, particularly an IMI project, and that's what happened. So how does an IMI project is being developed? IMI is an initiative of the European Union, and the specific partnership that was shaped is a partnership between the European Union and FBI, which is a group of pharmaceutical companies in Europe. And it works as follows. So let's say these are 6 companies that have an interest, for example, in autism, and they say, we bring together 50 million of in-kind contributions. So that's not in cash, it can be in cash, but in this case it is in-kind. And in-kind means that it is value that you bring to this consortium. So the 30 minutes that I speak here about is time that are not on projects within Roche, but actually work on this project, and that is in-kind contribution. When we bring 50 million in terms of the FBI partners bring to the table, then the EU says, well, then we bring 50 million in cash to this project. And together that would make 30 million. In this particular case, it will be 36 million because we generated some extra money elsewhere. So the project value is 36 million euros. EU Aims has 25 partners. That's what you see here. There's 36 million euro funding. Over the years, we employed about 100 to 150 scientists in 10 different countries. And the structure of the project you see here in the middle of this ring, where we have 5 work packages defined. So there's work packages on cellular assays, on animal models, on translational research, clinical research, and genomics. And it's all tied together by bioresource and management. And the senior leadership is myself and Declan. And if you want to know more, you can find that back in a paper in Nature, Drug Discovery. You can see more details of this project. Now, EU Aims is all about biomarker development, and particularly stratification biomarkers, so that we can divide the population and patients into subgroups that are biologically meaningful. In order to do that, you need to have data. And in the absence of data, so 10 years ago, as I tried to say in my first slide, there were no data. So we need to generate those data ourselves, and that's what we do in 2 different studies. We do that in one study called Babysitting Study, and the second study is the LEAP cohort. I will tell you a little bit more about these studies now. This is the first study that is a naturalistic observational study. It's called the Babysitting Study. And this study tries to identify early and predictive diagnostic markers. So it is a prospective study in infants at risk as compared to control children. How we try to do that? Well, we basically study families that have a first child diagnosed with autism and where the second child has a higher risk for autism. The infants that we study are ages 4 to 36 months. It's a multi-site study with clinical sites at London, Stockholm, Utrecht, and Nijmegen. So this is the first study. The second study is also a naturalistic observational study. It's called the LEAP study. That's trying to identify novel biomarkers for the stratification of cohorts. This is also deep phenotypic longitudinal characterization of children and adults ages 6 to 30. And it's also a multi-site study involving London, Cambridge, Stockholm, Utrecht, Nijmegen, Mannheim, and Rome. And these individuals are so controls and autism patients are very deeply phenotyped, and they're recruited in this study over the course of several months and years. So here you see the recruitment of this study. You see here in blue the projected recruitment, and in red the actual recruitment. And this is a stellar recruitment. On the right-hand side you see the protocol that these individuals have to go through, and it's spread over 2 different days, and it's a very deep characterization of these individuals. The LEAP study is a run under GCP standardization, which was very important for us because we wanted to use this study for building a regulatory framework in Europe. EU Aims was the first neuropsychiatric consortium that submitted a core protocol for quality advice on biomarkers, approaches, and methodologies. And we, in fact, had a very constructive 3-hour meeting with the EMA, which was very exclusive. I don't want to go in all detail about the recommendations that I have, but I will focus on 2 things. First, they emphasize the need for independent replication of our findings. And secondly, the caregiver-reported clinical outcomes should be supported by physician-reported outcomes. Uniquely, we then wrote together with the regulators a very nice paper summarizing our findings and discussions, and that was then published in Nature Review article. This paper then outperformed the average Nature paper with more than 3,000 page views in a very short period of time. We used that paper then to populate the regulatory guideline, which is still at the draft, but being mainly finalized, and also this is a unique document in the world that will help us develop drugs in this field. As I mentioned, one of the recommendations of the EMA was independent replication of some of our findings, In order to do that, we looked around in the world to similar initiatives like EU AIMS, and there were a few. So there's the FNIH network, Biomarker Consortium in the US, there is the POM network in Canada, there's a Chinese consortium, there's a French consortium, Fondation Fondamentale, and there's the CRC in Australia. And what we have done is reached out to these consortia and signed documents for data sharing and replication. The purpose of this slide is also to show that within this spider network, the center of gravity is EU AIMS. From the get-go, we decided to have a single data sharing platform, and this is currently situated in Institut Pasteur in Paris. And this platform is really also very exclusive. It will allow us to do data collection, integration management with access and search tools in order to accommodate all of the strategic studies that we want to do with the data that we generated within this project. And here are some of the examples that we could do. So there are now possibilities, for example, to do case control comparisons, correlation with symptom severity, technical validation, identification of subgroups. So these are all different studies that you can do, as well as a host of other studies. So this is really a fantastic tool that will bring us forward to some of the clear deliverables that we have in this study. And now I will give you one example. There have been a number of smaller academic studies that somehow were directed towards getting more understanding of the correlation of the relationship between anxiety and repetitive behavior. And these studies showed that anxiety leads to repetitive behavior, and that repetitive behavior then leads to an increased anxiety, sort of a self-driven anxiety repetitive behavior syndrome. Now the question here was, can we replicate those data in a large cohort like that for EU AIMS? And what you see here is the anxiety scores as a function of the repetitive behavior scores, and what you can see in individuals between 6 and 11, sorry about that. And what we clearly see is a very nice correlation, which is statistically significant. Now we're coming to the clinical network. These are all the clinical sites that we have identified. When we started, so it was completely empty. We did not know where centers of pediatrics were, where there was somehow a know-how about autism. We have mapped those now on this European map, and we have identified 96 sites in 37 countries. And it's one of the key assets, I think, of EU AIMS going forward. But what I really want to say is about recruitment, that we actually have now an idea of where these centers are in Europe, and this is a great result. What are these sites? Well, most of these sites are clinical research and universities that are somehow embedded into a larger structure. About 75% of these centers have patient registries in place, and that we could use, for example, for identifying patients that want to be part of the clinical trial, for example. On average, we have estimated that these sites together see about 18,000 newly diagnosed patients per year, which is quite a large population. And here you see the breakdown of age groups, with children being mostly diagnosed with autism. Then finally, there was an independent analysis by Reuters, looking at the portfolio of IMI projects, which are 59 within this particular call, and from various independent analyses, such as scientific achievements in terms of papers, etc., as well as contacts with key stakeholders, EMA and FDA. This IMI came out one of the top ranking, if not the top ranking project within that portfolio, which is an excellent result. And the last slide is really the people that have done all the work. The project is basically finalized now. There's 1 year of no-cost extension, so we have another year to go. But these are the people that have done the work. So thank you very much.
Okay, thank you, Will. Very nice overview. And we have now time for questions. So this is, of course, all meant to pave the way to testing new medications. And these new medications will be tested in context of moving towards precision medicine or personalized medicine. When do you think we will have new interesting compounds to be really implemented in clinical studies using all the preparatory work by the EU AIMS consortium?
Well, the pipeline of candidate drugs, they're in various companies. As far as I know, there's at least 4 or 5. One of them is actually in Roche, so we have one compound going forward in phase 3, which is it's in one of the later stages, and that would be available to the public in about, I would say, 6 to 8 years from now.
Will is talking about novel compounds, but I think one of the things to say also is that we've also been working with repurposed compounds to see if we can shift the dial on some of the biomarker differences that we've been finding. And in future webinars, we'll be talking about how by targeting the glutamatergic, the GABAergic, and the serotonergic system, we've been able to show that we can shift the dial on some measures of functional connectivity and also on glutamate and GABA concentrations in brain.
Yeah, that's great. So that would mean that you are repurposing, so you build those studies on existing medications, maybe for other indications, but then you build on safety profiles and other background data that are available. So this will allow for a more fast trajectory of testing these drugs.
That's correct.
Further questions, comments from participants to this webinar?
Jan, we got some questions in before the webinar started. One of them is, how does the outcome of the project refer back to its original objective?
Will, can you address the question?
What we will hopefully identify is factors or biomarkers that will allow us to address the heterogeneity of the population of the people with autism. As I mentioned in the beginning, this group is really heterogeneous, and you want to bring them together in terms of you want to create groups that are biologically meaningful. And with that I mean that have a more or less a similar underlying neurobiology for example, in brain function. Because that will allow us then to test certain new drugs in these groups based on knowledge that we have from the function in the brains that we then hopefully can address by certain new treatments. So the whole project is geared towards finding these factors that will allow us then to go forward and test some of our candidate drugs. Do you want to add something, Declan?
I guess the short answer is that we have hit all the key deliverables that we specified as part of the call. So we've achieved that. We are now undertaking the first large-scale data analyses after the cleaning has been done. And in upcoming seminars, you'll hear, for example, from Meng Chung, who you'll hear from next, how we've been finding that we can pass the heterogeneity around sex and gender. You'll hear also some upcoming talks from Eva Luch and colleagues talking about how she's been able to distinguish different subgroups using a cognitive profile. And you'll also hear in future talks how we're able to distinguish different subgroups based upon functional connectivity and anatomical connectivity and brain anatomy more broadly.
Thank you. Are there other questions already submitted before the meeting?
There were a few general comments where people are interested in gene discovery in autism and the clinical utility of genomic data. There was a question about any projects on gene discovery in autism.
I mean, there are several genes identified now as risk factors for autism. As I mentioned also, there's some familial traits for that. As far as I know, there are no specific genes that people are focusing on now for development of new medicines for autism, idiopathic for autism. However, for syndromic for autism, there are approaches currently going forward.
So it's Declan here. The point of this proposal was not to discover new genes for autism, but what it was, its purpose was to try and understand how genetic variation may be associated with variation in the clinical phenotype. And that work is being led by Thomas Bougeron. And the data for that is currently being analysed in collaboration with Autism Speaks. And so we can't speak directly to that at the moment. We hope that data will be coming through soon. I had a question from Matthias Dozer asking about how we ensured the quality of the diagnosis. And what we did in terms of people presenting is that they had to have a clinical diagnosis, and that was then validated, inverted commas, by use of the gold standard diagnostic and research diagnostic techniques of the ADR and the ADOS. Sorry, back to you, Amber.
So, Declan, was you addressing also the comment in the chat? There was a question from...
In the public.
About diagnosis and overdiagnosis. Yeah. It's maybe good to say that indeed we started with a clinical diagnosis, but we also added standardized high-quality research instruments like ADOS and ADI, and we will analyze the data both based on the clinical diagnosis as well as on the clinical diagnosis confirmed by clinical cutoffs on these research diagnostic instruments, which is a more thorough procedure. Amber, other questions submitted already?
We don't have any other questions that were previously submitted, so if anyone does have a question, please post this in the public chat. Perhaps it would be interesting to talk a little bit more about the tendency of overdiagnosis or self-diagnosis, and I believe we did not include anyone in the study with a self-diagnosis, but it might be interesting to comment on that.
Yeah, sure, and I would particularly like to ask that to Declan, because he has a strong record also working with adults with autism, and particularly in those adults, you have those adults that have never been diagnosed in childhood, and then in late adolescence or early adulthood are referred because there is some suspicion that they have autism spectrum disorder, or they are referred by their partner or by others. So Declan, how is your take into those, let's say, later diagnosed cases and the issue of overdiagnosis and self-referral?
Well, what we would classically find in a referral to a health setting of people as young adults who are self-diagnosed or are concerned they may have a diagnosis is in a tertiary referral health centre. We find that approximately 40% of those individuals actually turn out to have a diagnosis. In other words, 60% do not have a diagnosis of an autistic spectrum disorder. What they would tend to have would be a diagnosis of another personality disorder or other mental health disorder, such as obsessive compulsive disorder.
This is Matthias Dose speaking.
Yes, we can hear you.
Yes, we have the same experience with a clinical study which was done by the Max Planck Institute in Göttingen, and they also had referrals of adult people, and so there were approximately 60% in whom the self or diagnosis by psychologists did turn out not to be correct. So I think this is really an issue we should be very careful about.
Yes, I agree with that. And one of the problems, of course, is that we're lacking screening instruments which are reliable in healthcare settings.
Yeah.
And we have screening instruments for use in research settings that can distinguish individuals with, say, a high AQ score from individuals with a low AQ score, but those instruments, I'm afraid, don't work terribly well in clinical settings, at least in young adults.
Are you aware that a big group with several scientific communities, we created a guideline for the diagnosis of autistic spectrum disorders and are now working on a guideline on therapy, and we found out that we should recommend that if there is a suspicion of autism spectrum disorder, these people should be referred to specialized centers. So this may be one way to ensure the quality of diagnosis.
Thanks, Matthias. We had equivalent work in the United Kingdom that Tony Charman, for example, was involved in which they were setting out what they determined to be what we call the NICE guidelines over here.
Yes, we were aware of this.
Yeah. And so Tony was playing a leading role in that. I wasn't involved in that, but Tony and others, and Emily Sarnoff, etc. were. Okay, so if there's no other questions, I'd like to thank Will for presenting today and remind everyone that Meng Chung Lai will be presenting on the July 26th on sex and gender in autism. And thank you so much for joining us, and thank you all for listening in and to the organisers.